In this video, I'm going to discuss the roles of specific organelles in the process of protein synthesis. The first to discuss is the nucleus. The nucleus, as you know, is only found in eukaryotic cells, and it contains the genetic code for the organism. The genetic code is arranged on chromosomes, or DNA. So let's draw some chromosomes. Chromosomes are usually represented as X shapes. The chromosomes are the instructions for protein synthesis. They provide the blueprint for making proteins. Step 1 of protein synthesis happens on the ribosomes. So let's draw some ribosomes. Draw those in black. Ribosomes assemble the proteins by connecting amino acids. So let's draw some amino acids. We'll draw the amino acids coming off in a chain from the ribosomes. So these little blue squiggly lines represent non-functioning chains of amino acids, otherwise known as proteins. These proteins are currently non-functioning because they do not have the proper shape in order to function. Instead, the proteins are just long chains of amino acids. Step 2 happens in the endoplasmic reticulum. After the chain of amino acids, or non-functioning proteins, is created by the ribosomes, the protein is taken to the endoplasmic reticulum, more specifically the rough endoplasmic reticulum. So let's draw some rough endoplasmic reticulum. We'll do this in red. It's called the rough ER because when the ribosomes attach to it, it literally appears to have a rough or bumpy surface. The rough area of the ER is generally very close to the nucleus because the ribosomes don't carry the proteins far from the nucleus. So let's draw some attached ribosomes to the rough area of the endoplasmic reticulum. Now all of these ribosomes have their amino acid chains that they've put together. Now the job of the endoplasmic reticulum is to bend and fold these long chains of amino acids into the shape it needs to be in order to be able to perform its role. So it's going to take this long chain of amino acids, or protein, and bend it into shape. Remember, proteins must be a certain shape in order to function. Now, for the sake of this video, I'm going to say that the shape of our protein needs to be like a Pac-Man shape. There we go. So up in the rough area of the endoplasmic reticulum, let's go with rough. ER. The proteins are just long chains, but when they come out of the smooth area of the endoplasmic reticulum, they're actually bent into shape. Now these proteins are now molded into shape, and they are officially a functioning protein. They can go out and do whatever job they were designed to do by the chromosomes or by the DNA. Now, while the proteins are molded into shape and they can go out and do a job, they don't quite yet. They're not packaged up to be sent throughout the body. So what they end up doing is they exit the smooth ER and they work their way towards the Golgi apparatus, otherwise known as the Golgi body. So now I'm going to draw the Golgi here in green. So in the Golgi, the proteins are placed into packages called vesicles. These vesicles provide protection for the protein for their journey throughout the body or throughout the organism. The proteins work their way into these vesicles through a process called exocytosis, which I'll explain more here in a minute. The vesicles serve as a barrier around the protein to protect it until it reaches its final destination. So here we have this green Golgi body or Golgi apparatus. Now the proteins, while they're in their shape, go into the Golgi, and they'll eventually work themselves into these things, these areas called vesicles. So now what we have right here are functioning proteins that are in vesicles, or they're developing into vesicles. Let's show you what a final vesicle looks like. So I'll just draw a protein very quickly, and then I will draw the vesicle around that protein. So that vesicle is a protective barrier for the protein, because remember, it has to be a specific shape to function. So if it goes out into the organism or out into the body and in the bloodstream and it's getting bumped around, it could potentially lose its shape. So the vesicle protects that protein so that it doesn't lose its shape and therefore will be able to still function. Now, we talked a little bit about exocytosis. In the last part of the video, you saw that it starts to kind of like bulge out away from the Golgi body. But here I'd like to kind of describe that and show that in a little bit more detail. Okay, so we are now talking about the process of exocytosis. The exocytosis is how the protein, the finished protein, is packaged into vesicles. So for the sake of this video, or this part of the video, we are now in the Golgi apparatus, or the Golgi body. So this green line will represent the membrane of the Golgi body. Now, here is our protein that's in the Golgi. Now, the next step is for the protein to get closer to the wall of the, or the membrane of the Golgi body. So let's draw it much closer. Okay. Now, the closer it gets, the membrane will start to bulge out a little bit. So here, we'll go this way now. And you can see that this envelope or this vesicle is forming around the protein. So the next step, it'll bulge out even more. We'll put a protein in there. Oops, there we go. So the process of exocytosis, exo meaning to exit, right? So what's happening is the protein is exiting the Golgi body. And the last step, we'll have the protein here. There's the membrane, and now we have a complete vesicle. So it has exited the Golgi body through the process of exocytosis.